In this video, I want to explore the idea of microgravity, what it actually means, and why it's becoming both urgent and essential for plant science today. Over the past decades, plant biology has made remarkable progress. Those progresses advance our understanding of how plants respond to environmental stresses. Yet, as humanity pushes further into space exploration, especially when the SpaceX Starship completes its 10th test flight, it is now worth asking, should we begin to consider the future of plant science in space? Why raise this question? For a long time, plant research has often overlooked one critical environmental factor: gravity. Gravity is one of the most fundamental differences between space and Earth. On Earth, after hundreds of millions of years of evolution, plants have developed a finely tuned system of growth and development. For instance, plant roots always extend downward into the soil, while shoots grow upward toward the sky. Even if a seedling is placed horizontally, its roots can still reorient and grow in the correct direction. This phenomenon is driven by columella cells in the root tip. Columella cells are specialized cells. They contain dense, starch-filled organelles known as amyloplasts. These amyloplasts sediment in response to gravity. They enable the root tip to sense direction and align its growth accordingly. In space, however, this gravity-dependent growth mechanism no longer functions properly. This is not the only impact that plants grown in space will have. Similar challenges arise in other physiological processes. For example, plants use light and carbon dioxide to produce oxygen and water, but in the absence of gravity, water vapor and oxygen may accumulate around leaf surfaces, potentially blocking stomata, the most important respiratory organ of plants. The biosynthesis of plant-specialized metabolites will also be affected. Many specialized metabolites are synthesized through multiple cell types, which means a lot of transporters are required to shuttle intermediates between cells. The absence of gravity in space is likely to affect the communication between cells. This probably affects the synthesis or accumulation of specialized metabolites. However, we currently lack substantial research on this topic. The absence of gravity may also affect the interaction between plants and microbes. Microbes require direct or indirect contact to infect plants. For example, after a virus infects a cell, after propagation, virions spread to surrounding cells through the plasmodesmata, the tiny channels between cells. Would the spread of viruses be affected in space? We don't know. All these questions reveal an important gap. Much of the knowledge we have gained from Earth-based plant research may not fully explain or predict plant behavior in space. It may be time to treat gravity as an additional abiotic stress factor in plant biology. This does not mean that we need to establish a space plant laboratory immediately. At present, building a dedicated space laboratory for plant research is not yet practical. However, ground-based facilities like the 3D clinostat can solve the problem by eliminating or averaging out the directional influence of gravity. While these methods cannot completely remove gravity, those machines can create a simulated microgravity condition. They allow us to collect preliminary data on how plants respond to microgravity. It also makes it possible to study the plant response to microgravity using state-of-the-art technologies such as single-cell omics, spatial transcriptomics, and proteomics. We can begin to unravel the mechanisms of plant adaptation to microgravity, laying the foundation for future studies in real space. The visuals and materials you'll see are gathered from the internet or created with AI. I'd like to thank all the original creators. Their work helps us share knowledge and make the world better.